Okay, so let's go again. Are we good to go? Yes, okay, super. So welcome everybody, and thank you so much for the invitation from the organizers of this workshop. It's an absolute pleasure to join your program today. And I think the topic that you've selected is highly suitable and really interesting, and it focuses on the child as an active agent in their environment, constructing their own learning experiences. And we know, of course, that one of the most important sources of inspiration and interaction in the early environment are other adults or other humans. And one of the most important people whom the child would be learning from is their parent. So it is the parent-infant relationship that we study in my lab, and we look at how those social interactions potentiate early learning. And we're particularly interested in the neural processes that underpin this. And so we employ what we call a dyadic neuroscience perspective, which involves concurrently collecting brain signals from two interacting partners. As you will see on the slide on the right-hand side, typically it's a mother and a child who comes into our lab. And we fit them out with these soft, stretchy EEG caps, which are like swimming caps. And these sensors are able to give us a readout of the ongoing electrical activity that's occurring while the mother and child are talking together, playing together, or engaging in other forms of social interaction. And we've been able to glean some very useful insights into the learning process using this novel technique. Before I go into that, a little bit more about myself. I do hold joint affiliations. I'm an associate professor of psychology and medicine at NTU. I'm also an honorary senior fellow in the pediatrics department at Cambridge University. But as one of my enduring interests, I'm also a deputy director of the Cambridge NTU Center for Lifelong Learning and Individualized Cognition. And this is where we look at how these learning processes change or are moderated over the lifespan. And so we also study adolescents, older adults, etc. And we focus on executive function skills, particularly cognitive flexibility. But for today's talk, I will, of course, focus on my infancy work. So why do we study the parent-infant dyad? Well, in terms of their dynamics, there is a huge amount that goes on between the parent and the child. And in fact, you have to view them as one interconnected ecosystem, because what the parent does intrinsically affects the child and vice versa. And they're highly, highly connected at multiple levels across multiple domains. And this kind of level of connectedness can be broadly described in a phenomenon called biobehavioral synchronicity. So what do we mean by synchrony? It's not necessarily the case that whatever the parent does, the child does, or vice versa. It's more the case that they share a predictable pattern of change in behavior. And the best way to describe this may be to draw an analogy with doing a dance. So when you see two people dancing the tango, for example, they're not necessarily moving their legs in the same way at the same time or moving their hands in the same way at the same time, although they might. It's more the case that their actions are closely coordinated. So if one partner steps forward, the other partner steps back to accommodate. If one partner issues an invitation, the other partner responds by taking their hand. And so there's this give and take, this rhythmicity and flow, and they work together in one connected unit. But they kind of follow the same time frame of coordination so that their actions become predictable one by the other. And that's the kind of synchronicity that we're interested in here, this high level coordination of behaviors, but also of underpinning neuro and neurophysiological systems. So in my lab, we study these synchrony states, states in which we can see high levels of coordination and mutual predictability in patterns of change. Now, I should also say that these synchrony states are not something that, you just be in synchrony and you're in synchrony for hours, although that might be possible. What we usually observe is that we drift in and out of periods of synchronicity, and these are transient states. So you may be highly in synchrony for about, half a minute, and then that state decays and you naturally drift apart again, but then you reconnect and you drift apart again. So these are transient states of connectivity or synchrony that seem to be useful or serve some important cognitive processes that we have been studying. So one of the interests in our lab is now that we see this natural ebb and flow in levels of synchronicity, we can see these states being created and then dissolving. What are the cues or the conditions and the context under which parents and children establish synchrony? So how do we get in sync with each other in the first place? And what our work has shown us is that there are a large number of social cues that we can use to trigger or attune to each other. And actually some of them you probably have already heard a lot about. Unsurprisingly, one important social cue for synchronicity is eye contact or gaze. That's maybe one of the reasons why, when we're doing these talks, it's a little bit harder to get in tune with your audience because you can't look them in the eye, or you can't be sure they're looking at you in the eye. Whereas when I teach in the classroom live, I can look at each student and make sure that they're following me and that they're on board. And in fact, we can measure that effect neurophysiologically. I'll show you the data later, that when you lock eye contact with somebody, your brains naturally go into a state of mutual synchronicity, and that state seems to be quite beneficial for communication and learning. So gaze is something that is a synchrony cue and that we spontaneously use during the course of social interaction to establish temporary states of synchrony with our partners. Unsurprisingly, speaking also helps to establish synchrony. And I hear you have other talks on language as well. But there are particular patterns of vocalization, for example, infant-directed speech or using a child's name, that seem to have a similar effect in establishing a temporary boost in mutual synchronicity. And finally, we've also looked at emotion as a modulator of interpersonal synchronicity. And what we found is that when we share states of positive, happy emotion, that also naturally connects us to other people. And in that connected state, it almost is the case that we're operating as one unit rather than as two separate units. I may not have time to go into that today, but there are papers that we've published on that dimension as well. So there are a range of social cues, and I don't think that this is actually exhaustive. I think actually social behaviors are evolved to be very effective for connecting us to other people. So I think this may be just the tip of the iceberg for how, in fact, we do establish contact and synchronicity with other people. The other dimension, which I've been very interested in exploring, is touch. How early contact, skin-to-skin contact, or other forms of touch are also equally powerful as synchronicity cues. So I think as the field matures, you will find that there are more and more patterns of behavior across all our sensory domains that may be effective in establishing synchronicity with other members of our species. So that's the how. But also we are interested in the why. I've alluded to this a little bit. It's not just something that's a curious phenomena that happens, kind of like as an artifact of the processing that we do in EEG. We've shown that you can demonstrate synchronicity over and above whatever you expect by chance, even in two people who are experiencing very similar environment. There's something over and above that, which is quantifiable as synchronicity. So what does this do? Well, we've studied the effect of synchronicity in several different conditions, and we've seen relationships to communication in that it supports the child's efforts at communicating back to the adult. And more pertinently for today's talk, it supports learning. It seems to potentiate what we call a learning-ready state. So coming into synchrony with each other isn't just a pleasant experience that helps us bond, although, it definitely is the case that when we feel like we're connected to somebody else, that's a very pleasant experience which helps affiliation. But it seems to have cognitive positive effects as well in supporting communication and helping us to learn from other people. So how do we study and quantify these states of synchrony and the mechanisms that underpin them? So we use a method that we call dyadic EEG. Our colleagues also call this hyperscanning. Hyperscanning is the more general term for imaging of two or more people, and it can be done using MRI, using FNIRS. Our preferred modality is EEG, it's simply because it's low cost and it's easy to use with interacting partners. And so typically we have mothers and infants, and we collect these EEG signals at the same time from both of the partners. We get these time varying signals, for example, you see there in the screen, and then we compute causal indices to establish the strength of the influence of one partner on the other. So you can see the arrows there in blue indicate, certain sensors, or it could be certain brain regions, and we can compute their directional influence from the mother to the infant and vice versa. And to this kind of directional computation, we usually have to use more advanced metrics. So we go beyond simple correlations, for example. We use more advanced modeling metrics like Granger causality, and one of the indices that we like to use is called partial directed coherence. And this is a direct and a directed measure of interpersonal connectivity. I say direct and directed because those two are meaningful terms. So when we say directed, it means that we can pick apart the the causality here, so the direction of information transfer, or the direction of influence. So we can separately compute the influence of mother on child separately from the influence of child on mother. Now, in a method like correlation, you can't pick those two things apart, which is why you can't usually infer causality. But with a method like PDC, you can, because you do time series modeling to look at the extent to which one time series predicts the future of the other. It's also direct because it takes into, it can isolate the specific influence of, let's say, sensor A in the child on sensor A in the mother, independently of any indirect influences through sensors B or C or D. And so it is really a direct and directed measure, which is why we found it quite useful in our methods. And we can talk more about methods later if you like. Okay, so just an overview of the points, the major points that I want to cover in my talk today. So I want to elaborate a lot more on how social interaction supports early learning. We know there's plenty of evidence for this already in more general and certainly behavioral terms, but of course I'm interested in the neuroprocesses that underpin this. So exactly how does social interaction support learning? And then we want to look more particularly as a function of this mechanism into the role of these social ostensive signals, which I have indicated to you may have a particular role in aligning or tuning brains into a state of synchronicity. And then I want to talk a little bit more about what we mean by learning readiness and what happens when you are synchronized with somebody else. And then finally, I want to zoom out a little bit into exploring some avenues for translation or application, and exploring the idea of using these social interaction indices to look at the quality of interaction, of course, but also to look at whether these can predict the trajectory of a child's brain development. So first of all, how does social interaction support early learning? So I think it comes as no surprise to students of psychology that the human species is highly, highly adept and indeed maybe adapted to perform social interaction and social learning. In fact, it has been proposed that our mastery, our particular skills in engaging in social relationships, in particular second personal social relationships, is one of the key factors in why we have become so successful in our evolutionary journey and become so dominant. And the idea is that we can accrue cultural intelligence through this means. So the difference here between social or second personal learning and direct learning really is that you don't need to have direct experience of the object or the knowledge to be able to learn about it. So direct experience, for example, maybe in caveman days, is when you come across a bush, right, in a forest, and you've never seen this bush before, and you want to learn about whether these odd-looking purple berries, for example, are nutritious and good for you, whether they're poisonous. Now, there are two ways to learn or to acquire that information about whether something is poisonous or not. There's direct learning, which is you take the berries yourself and you put them in your mouth, and then you eat and you swallow, and then you wait and see what happens. That's direct experience, right? And you can learn about it very effectively that way, because if you survive and it tastes good, you've learned, right? Those purple berries are good for food. Unfortunately, this also carries a huge risk to yourself, because if those purple berries were poisonous, you also carry the risk of being poisoned by them, and you may not live to carry the tale. So there's a second way of gaining the information about the poisonous or good state of the berries. You can ask your friend. You can say, hey, Tom, come over here. Why don't you try those berries and see what they're like? And then you can fold your arms, stand at the side, and watch while your friend eats the berries. And by observing his facial expressions, by observing whether he clutches his belly and rolls over, and observing whether he froths at the mouth, you can gain the exact same information that you need, which is, are these berries good or poisonous? But you do not risk anything to yourself, because you learn by watching. Now that is social learning, or learning through second-hand or second personal experience. And it is that form of learning, that second person appropriation of information through other people, through watching, through communicating, rather than having to directly step in and experience things yourself, that we are so successful at. So that was a caveman example of berries, but actually we are doing that right now. I'm telling you all about dyadic EEG, and I'm gathering not many of you would have actually stepped into the lab to do dyadic EEG, but now you can talk with some level of confidence to somebody else about what dyadic EEG is. So you've also employed this form of second personal learning. So really, it's the human species' ability to do this, to appropriate information through indirect means, through indirect learning, through watching, observation, and communication, and of course, interaction, that has allowed us to do so well in our evolutionary journey, reduce the risks to ourselves, accrue what we call cultural intelligence, and shared knowledge. And in fact, social media is a perfect example of that as well. There is a collective intelligence of the social media hive, which we can tap into and learn from anytime we need to know about what's going on in the world. So classically, when we study developmental psychology, there are a few common mechanisms that come up when you ask, how do children learn through social interaction? One of them is joint attention. So this is where the child shares the focus of their attention with an adult. And it also typically involves an object or an event. And we know that these periods of joint attention typically predict learning afterwards. There's also another form of learning which maybe has received a bit less of a spotlight, mainly because it's thought of to be no more than maybe simple imitation, but actually it's more than that. So this is the form of learning that I want to elaborate on more here, and that's called social referencing. And the idea here is that the child is able to use information gleaned by watching an adult partner in order to inform their own understanding of a situation. And this information that the child watches and learns about vicariously can either be information about actions, and so this is what we call instrumental social referencing, or in the classic Bandura literature is what we call observational learning. So there, the adult hits the Bobo doll and the child learns to hit the Bobo doll. So it is a very simple kind of instrumental imitation form of learning, where the child watches an action and replicates the action. But the child can also learn from facial emotions and from the adult's appraisal, social appraisal of the situation. And in that case, it's not a simple one-for-one action imitation. It's more complex than that. The child is using the adult's appraisal or emotional responses to a novel object or to a novel event to frame their own approach or their own understanding of the situation. So that's a more complex form of learning, which is pretty powerful. And actually, it's the basis of advertising that people also use when they try and sell you to try a new product. So one of the earliest demonstrations of this, you can see on the right-hand side, was using this visual cliff apparatus. Now, those of you who are students of developmental psychology will know this visual cliff apparatus was originally used to study depth perception, but as it turns out, it's also quite useful for studying affective social referencing. So in this paradigm, the child is put on the far end of the apparatus and the mother stands at the opposite end. And the child sees the mother and tries to crawl towards the mother. But halfway across their journey, they reach the edge of this visual cliff. Now, to the child who has developed depth perception, it looks like there is a very sharp drop off in the middle of the table, like the table abruptly ends because there's only glass after that. They don't know that, of course, it's the glass. It just looks like there is a very sharp cliff. So it looks dangerous. So at that point, the child usually freezes because they're uncertain. They don't know, is it safe to carry on? They don't know what to do next. And that's where affective social referencing kicks in, because the child will look to or reference the mother on the other side. And if the mother is smiling and reassuring and positive, the child is much more likely to take the leap of faith and continue crossing the cliff over the plexiglass portion. However, if the parent is fearful and negative and worried looking, the child will take that as a danger signal and freeze and usually either crawl back or just become incapable of moving forward. And so it's very clear that in that situation, the child's behavior in that period of uncertainty is shaped or formed by the parent's appraisal of the situation. Is it safe? Is this something you should go ahead with, or should you stop and question your decisions? So children are capable of doing this from as early as 12 months of age or even younger. So social referencing of this form develops quite rapidly over the first year of life. And this is the type of learning, this type of social referencing or learning through other people's emotions and appraisal that we are very interested in studying in our lab. And when we study social interaction, I think we cannot get away without studying play, because most of our interactions with young children are highly playful. Only rarely did we put them in these semi-precarious situations that you saw in the cliff experiment there. Most of the time, when we interact with our children, the most natural way that we interact is to play with them. or to sing and talk and have nursery rhymes and read stories is usually enjoyable and pleasurable. And so one of the first things we wanted to do was look at these learning mechanisms in the context of a very warm, friendly, interactive play scenario. So I'm going to talk you through now a series of studies where we started with this play scenario, and then we worked our way onwards towards the more formal social affective referencing type paradigms. So this is one of the earliest ones that we did back in 2016 or so, when I was collecting the data in Cambridge. So you see British mums and babies here. And we started with just a fairly small bunch of parents and children. We then added more data later, but these early papers were published just on this initial cohort. On average, the children were about 11 months or so. And what we did was we asked them to play together, which we call joint play, or separately. So in the joint play condition, both the mother and the child shared the same object, and they were asked to play together. And so the mother naturally would engage in eye contact, and she would be manipulating the object with the toy. She would be manipulating the object with the child, and they would be sharing their attention. The mother would be trying to bring the child into the activities and interacting with her. In the solo play condition, they were sitting in the same condition, but instead of playing with one object, we gave them two objects, but the objects were the same. So they each played with their own toy, and the toys were identical. But we created that sense of individuality by placing a visual barrier across the table. And this barrier, as you can see on the far right picture, was carefully calibrated in height. It was tall enough so that the child, when looking upwards at the parent, couldn't see the mother's eyes. The child can just about see the top of the mother's head, so they know the mummy's there and they won't feel insecure and worried that the mother has left the room, but they can't establish eye contact with the mother. And so it feels more like an isolated experience. So that was the solo play and the joint play, and every dyad did joint play and solo play. And then we coded for the visual attention of the child and the parent. Of course, we're very interested in the child particularly. And we looked at where the child was looking. Was the child looking at the object, the toy, or were they looking at the partner? And these proportions of visual attention differ during the two different conditions of joint and solo play. Now, we also collected EEG. This isn't shown in the illustration, but the parents and children were also wearing our EEG caps. So let's start with this first basic hypothesis, which is looking at how the locus of attention changes as a function of social interaction. Now, I've asked you to predict a priori, what do you expect to happen differently when you're playing with your own toy versus when you're playing with a parent, then I think most of you would probably say, well, there'll be an increase in the amount of shared attention with the parent in the joint play condition, because there's an opportunity to share attention. That's the most obvious hypothesis. And in fact, yes, that's what we found. There was a slight increase in the shared attention with the parent, 0.05, 0.052. But actually, that wasn't the major effect. What surprised us more was that the biggest effect was in the amount of attention that the child placed on the toy when they played with their parent. So you can see that those looking times on the left-hand side chart there, where the blue shows joint and the orange shows solo. The three categories that we coded were looking to the object, looking at the partner, and being distracted and looking elsewhere. So the main effect of looking to the partner is shown in the middle section of bars. So yes, there was a small increase in looking to the partner during joint play. But as I said, the biggest effect that we observed is that when parents and children play together, the child spends much more time looking at the object than they would if they were playing with the object just by themselves. So it is not the case that there's a trade-off, that just because you're looking more at your parent, you're more distracted or you're not paying as much attention to the toy. Rather, it's a positive boost in both directions. When you play together, there is a social benefit, but there's also an attentional benefit on sustaining attention on the toy, the focus of attention. And the only thing which seems to drop is inattention. So when you're playing with your parent, the child is less distracted. They're more focused. They sustain attention for longer. So that's something which we kind of didn't expect, but which turned out to be quite important in understanding the dynamics of social interaction and why this helps learning. So as I mentioned, we also collected the EEG from these children. And in the second paper, the PLOS biology one, we looked more specifically at the neural effects that were explaining why the child was sustaining attention for longer on the objects. So in this analysis, what you see here is a cross-correlation plot that's looking at changes in parental EEG power across all the different frequencies. So the maternal EEG frequencies are on the y-axis there, and we've plotted them so that the lowest frequencies on the top and the highest frequencies at the bottom. So the different rows are changes in maternal EEG power, and we've cross-correlated that with baby's visual attention. And so on the x-axis there is correlations with the child's looking behavior. And what we've done is a lagged correlation. So it's a cross correlation to look at whether maternal changes in brain activity predict, so they come before, or follow, which means they come after, changes in baby's visual attention. And the colors on the plot show you the strength of the correlation. So the hotter the color, the stronger the correlation is. On the left-hand side, you see the cross-correlation plot for playing by themselves, and on the right-hand side for playing together. And just visually, you can already see there's a lot more going on the right-hand side, which is the joint play, which means maternal changes in brain power are a lot more closely related to what the baby is doing in terms of visual attention when they're playing together. That's not surprising. But of course, we have to apply the appropriate thresholding and statistics here. So we did a bootstrapping analysis to check whether any of these changes were statistically significant, and actually none of the relationships were significant in the solo play condition. But in the joint play condition, there were these bands of changes where maternal brain activity did predict baby's visual attention. And that happened specifically in this range of about 3 to 6 hertz there of parental EEG, which is kind of in the theta, so very, very low alpha, but mainly theta range for the mother. And what we found there is that changes in this kind of maternal theta neural power were predicting baby's visual attention behavior. But the more important thing actually was the direction of the prediction. So I mentioned that we're very interested in directions of causality here. Is it brain changes predicting visual attention, or is it, baby looking and then mother's brain changing as a consequence? And actually, if you pick those out, so this is kind of like a flat summary version, but these are actually strengths of correlation, which you can then plot on a plot here. So this shows you the same thing. In the solo play condition, there's no significant association. So the line there is very flat. In the joint play condition for this EEG band, you can see this huge increase in correlation in the cross-correlation plot, which is statistically significant. But the important thing is, where does this peak happen? And actually, without boring you too much of the details, the peak of the correlation plot comes on the right-hand side, which means that in this case, it's the maternal brain activity following changes in baby's attention. So it's not the case that maternal brain activity comes first and then babies look after. It's babies looking at something, mommy's brain is responding to that. So it's responsiveness that's significantly coming out as dynamic in this scenario. And to take this one step further, so just to hammer home the point here, the direction here is that maternal brains are responding to babies' changes in visual attention. And just to close the loop back, the more responsive the mother was, so individual mothers, as a whole, all the mothers' brains responded to changes in visual attention in the child. But there were individual differences, and also there were differences across different instances, different epochs. And what the right-hand side plot shows you is that the more responsive the mother's brain was, the longer the child sustained their visual attention for subsequently. So if there was basically, it works like this: if the child has decided to pay attention to an object and has shifted their focus of attention to the toy, the maternal brain responds to that. And the more responsive the maternal brain is, the longer the child subsequently keeps paying attention to that toy. So the responsivity has the effect of prolonging or sustaining infants' attention once the child has allocated their attention to that toy. So it's really a sustaining or a scaffolding effect that we're seeing here. So that's the dynamic that I wanted to show you, which is, what's actually going on in the micro ecosystem of mother and child when they're playing together, and what are these cognitive benefits? So we've seen one of them particularly is in sustaining, prolonging joint attention, and to be able to then use that period to learn about the object that you're focusing on. The other thing that we wanted to study was these signals themselves. So we had an idea that, eye contact, changes in visual attention, something is going on here that involves looking. So there must be something powerful about shifts in gaze, which are tuning or bringing people into synchronicity. So we thought about this more broadly to begin with. We thought, okay, there are probably a class of cues that have this function. And actually, classically in cognitive psychology, you would probably know that there are these ostensive signals, which are there to convey our communicative intent to a child. And basically, they're saying to the child, I'm here, I'm online, I'm ready to connect with you. This information is relevant for you to learn. And there are some key features of these classes of signals. They unambiguously specify that the child is the correct addressee. They're very easily discriminable, so they're not vague at all. They're very obvious, perceptually salient cues, and they work. When you use them, like when you use a child's name, they induce the orientation of the child towards the source. And we know that at least there are these three types of signals which fit into this category, and they're eye contact, using the child's name, and using infant-directed speech. And in fact, just a small digression, but we also know that in some cases of maternal mental health disorder like depression, there is a drop in being able to use these cues effectively to engage with your child. So mothers who have depression, for example, are less able to engage with their child using ostensive gaze, and their use of infant-directed speech is also more flattened. And that kind of goes along with the reduced responsivity and contingency profile that we see in this class of patients. Okay, so ostensive signals are very interesting for us to study, and we did come up, or we thought about how these might actually work to align brain signals to each other. And so our thinking, if you have the time to read through this, was laid out in a TICS paper that we published last year, and that was to explain mechanistically in terms of brain signals what these gaze cues and other cues might actually do when they are given and received. So I'm sure many of you are aware that when we collect EEG, we're looking at these ongoing oscillations in the neuroactivity of the individual. And this oscillatory activity can be phase reset, or it can be aligned so that the parent and the child's oscillations become mutually predictable. Now, in this graph here, I'm making it very, very simplified, which is just to have the most simple model of synchrony where the oscillations are basically perfectly in time and perfectly in phase. And so that will be a case where if the child's oscillation is in a peak, the parent's oscillation is also in a peak, and vice versa. And so you see exactly the same pattern. But as I explained at the start of this talk, that's not the only model of synchrony. You can also have synchrony where parent is in peak, child is in trough, and they just go anti-phase like that. Or you can have leader-follower relationships where parent is in peak first and then child becomes in peak, and they follow each other at a lag, but it's a consistent lag. Or you can have other more complicated models of synchrony where you have the parent's oscillation speeding up a little bit first, and then the child speed up, and then the parent's oscillation slows down and the child slows down. So there are many ways in which you can describe synchrony as a fairly broad phenomena, but this is just the most idealized and simple case to understand. So what we're proposing that one of the ways by which you see this increase in synchrony may be by a point-wise adjustment in the phase, instantaneous phase of one of the partner's oscillations. Because in the absence of anything synchronizing us, our oscillations have their own internal timing. We have our own internal mechanisms which are clocking us. So we're each oscillating on our own time in our own phase patterns. But something happens when we connect so that one partner adjusts to the other. And one of this adjustment mechanisms is something called phase resetting. And we've seen this happening. So we know that our oscillations phase reset. In the classic visual neuroscience and auditory neuroscience literature, we know that phase resetting happens to salient cues. So when you see a very bright light, for example, or when you hear a stressed syllable that's very loud, or when you hear your name, those are sharp, salient events in your auditory or visual environment that induce this kind of phase setting response. Your ongoing oscillations shift and reset their phase. Why do we need to do that? I'll talk a little bit about that later, but this is one of the possible mechanisms that we think happens, which is that when an adult uses that cue, like looking at the child or saying the child's name, this is a salient cue that phase resets the child's ongoing oscillations so that they are then after that coupled and in synchrony. So I'll come back to this question now of why do we need to do this? Why is this phase so important? And on the top right-hand side here, we've got these bars in green that I've called high receptivity. And that really is the key here about why this oscillatory synchrony is so related to the mechanism of learning. Because these oscillations, the peaks and the troughs relate to periods of responsivity or receptivity in the brain. And the idea is that in certain oscillatory phases, classically it's the trough phase actually, that is when the particular population of neurons that you're studying is receptive to incoming information. So it is collecting and aggregating information in that time window. So it's receptive. But then when the oscillations change into the peak phase, for example, that is a non-receptive phase. So that population of neurons is no longer actively receiving information, it's actually sending information on to higher order centers. And this model has been proposed for the early sensory cortices at least, that there are certain phases in the oscillation when we're actively collecting information about the world, and there are other phases when we're not really outwardly facing the world, we're processing and passing the information on to higher order centers. So the information that comes in during the peak phase in that sense, when we're non-receptive, actually gets missed. But we don't see this, we don't feel this because we're very good at interpolating. So for us, vision and consciousness feels like it's continuous, but actually that's an illusion. We are actually interpolating, we're sampling our environment very, 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 very rapidly, and we're stringing together these individual slices that we sample. And the analogy there is like viewing a movie. When you go and see a movie, what you're actually seeing is many, many, many picture frames being flashed in your eyes at a very high rate. But when you experience it as continuous because you interpolate from one frame to the next. And indeed, our attention sampling of the world is very similar. We have periods where we chunk and sample information from the world, and then we interpolate between them. And what the phase resetting does is it makes sure that the parent's peak responsiveness is aligned to the child's peak responsiveness, so that if the parent is passing information on, that hits the child's most optimal receptive period. And that is the essence, I think, of why synchrony is so important for encoding and therefore for downstream learning. Okay, so that's a theory. But it's just a theory. We don't know if it's actually true yet. But we've started to run some studies to give us some insight into this. And so this is one of the first ones that we did, which was published way back in 2017. And it was testing the effect of just one of these social cues, eye contact. And we had a very simple hypothesis that when you look the child in the eye, you're going to increase brain-to-brain synchronicity with her. So we designed two conditions. Sorry. So the babies actually took part in two conditions here. One of them was interacting via video. So actually the stimulus there was pre-recorded, and the other one was live. In both cases, we had the same adult. So the same adult was either pre-recorded on a video or she sat there in the lab interacting with the child. And the paradigm that we used was nursery rhyme singing. So we had the adult singing, the wheels on the bus go round and round. But that was really just the carrier for the stimulus. The thing we were interested in manipulating was eye contact. So the experimenter sang to the child, looking directly at the child, which we call the direct condition, or looking indirectly, which means looking 20 degrees to the side, but she was still singing. So the auditory stimulation we matched very closely, and those sung nursery rhymes in the different gaze conditions were very, very similar, statistically indistinguishable. But the difference was whether there was eye contact during the singing or not. So we measured the brain activity of the adult and of the child. And what I'm showing you here is the key take-home lesson, which is in the live condition, but we saw the same thing in the video condition as well. And we were able to collect brain activity from the adult and the child at the same time. And what you see here are the directions of connectivity, which were significant in those two conditions. So I mentioned that using measures like PDC, we can pick apart influences of one electrode on another electrode very specifically. So that's what these arrows represent. We can look at information flow or causal influence from the infant's right electrode to the adult's left electrode, et cetera, et cetera, et cetera. So each arrow represents one possible connection. Now, if the arrow is there, it means there was a significant connection after we have thresholded and removed anything spurious, controlling for environmental interactions. And the thickness of the arrow tells you how strong that influence is. And so we can see that there was a lot of influence coming from the adult on the child. Those are the big, bold red arrows. But of course, the strength and the size and number of these arrows is much stronger in the direct condition than in the indirect condition, because the indirect condition, we got much fewer of these significant connections and flows going on. But interestingly, in the live condition, we also had influences coming back from the infant to the adult, and these are the blue arrows that are coming back. And again, in that direction, there was a stronger influence of the infant on the adult, also in the direct condition. So when we summarize these arrows, we take the grand average of them. That's how you get these bar plots at the top, where you see on average, if you look just at the flows of adult to infant and vice versa, there's a very big main effect of eye contact, direct or indirect, on the strength of connectivity between the adult's and the infant's brain. And then just to close the loop, we also measured the child's communications back to the adult. So here's the adult singing nursery rhymes and, you know, trying to engage the child. Was there anything coming back on the other side? That's what we were interested in. So we measured this. So we measured the child's vocalizations and attempts to communicate. And we found an interesting relationship, which is that the stronger the influence of the infant on the adult, that's these blue arrows coming back. Each of those dots is one child there. The children who had a stronger influence also were the ones who were making much more of an effort to communicate. So the ones with the strongest influence on the top right-hand side also vocalized for longer, for a more sustained period in trying to communicate back to the adult. So you can see here that synchrony was part of this mechanism of maintaining the communication within the dyad. And that, yes, the adult is actively kind of communicating to the child with these nursery rhymes, but the child also has a reciprocal role in sustaining the loop by vocalizing back. And that vocalizing is also driving this feedback synchronizing coming from the child back to the adult. So it's a bit of a virtuous cycle there, where the adult is offering these communicative signals and synchronizing the child, and the child is responding back with communication, which is also synchronizing the adult back. So you can see that this dyadic ecosystem is being set up in this case, and it's obviously much stronger during direct eye contact than during indirect eye contact. So this relationship with vocalization did not exist in the indirect condition. We only saw this when there was direct eye contact. Okay, so we've kind of seen one of the snippets or the vignettes here that, you know, eye contact does have this immediate role in kind of triggering and generating a transient state of synchronicity and maintaining this feedback loop. But does this actually potentiate learning? So we come now to the last set of my experiments that I want to talk to you about, where we specifically look at this affective social referencing. So the context here, like I've explained already, is the idea that when your brains are in synchrony and your receptivity periods are aligned, it's easier for information to be transmitted from sender to receiver. Basically, the infant is ready to listen when the adult is ready to speak. But we need to test this hypothesis more specifically. So we did our own version of the affective referencing paradigm, but instead of doing the visual cliff, because that was slightly health and safety worrying, we didn't want babies actually crawling around on plexiglass surfaces on a raised platform. So we did something with toys instead. And so this is also a paradigm that's been used to study social referencing. We give the parents a pair of novel and very ambiguous objects. So my postdocs went to a DIY store and got some bits of, you know, piping, for example. And the idea of this is that you don't want the child to have any prior knowledge or any preconceived notions about the object. So they have to be completely novel, which then induces a state of uncertainty. I've never seen this before. What are they for? Are they dangerous? Is it good? Okay, so these were pairs of novel objects that we gave to the parent, and then counterbalanced, we told the parents to say, look at one object and express positive emotion towards it. Say, oh, this is really great. This is fun. I like this one. And then look at the other one and go, ugh, this is yucky. I don't like this. Ugh. So to give very clear social cues and references as to one which was positive, approved, liked, and the other one which was disgusting, negative, disapproved. Okay, so this is what we call the observation phase. Baby watched while mummies picked up these objects and displayed these interesting emotions towards them. And then we offered both objects to the child and we measured their response. Which object did they choose to play with? Now, if you're a normal parent, like most of us, you would want to see that the child goes for the object that you approve, right? So if mommy says, oh, this is great, you would love to see our child going, yeah, I'm going to pick that one up and play for it. If mommy says, ugh, don't touch that, you want to see that the baby avoids that, right? So this is a very practical situation, actually. And there are a lot of real life analogies to this. For example, if you're trying to get your baby to try a new food for the very first time, like broccoli, they've never seen this before, you know, to be fair, they won't want to try it because they don't know what it is. So what do parents do? They do something like this. They pick up a piece of broccoli, they put it near the mouth, they make a big show and tell of it, they eat a piece of broccoli themselves and they smile and they go, oh, this is yummy. And we hope that the child will watch our facial expressions and learn that broccoli is yummy, and then they will eat it themselves. So this is a very close analogy to that kind of very practical real world situation. Okay, so coming back to the science of it now, what do we actually measure? So during the observation phase, we collected the brain signals from the mother and the child at the same time. And this is the period that we're most interested in analyzing. What's going on during this learning referencing period? Okay, so we were able to compute connections within the child's brain, within the parent's brain, and of course, between the parent and the child, every possible pairwise connection there. And we wanted to see which of these patterns of brain connectivity predicted the subsequent response of the child. And that was during the response phase. And here's where I need to spend a bit of time explaining to you how we defined learning in this paradigm, because it's not as straightforward as you would think. Now, you and I would think if the child goes for the positive object, she's learned. If they don't go for the positive object, she's not learned. But actually, it's not as simple as that, because it's entirely possible for the child to consistently go for the object that you say is nasty or negative. There were, in fact, 1/3 of the children in our cohort who did that. The mothers say, this is great, and the child goes for the one that the mother doesn't like. Now, are you going to tell me that those children didn't learn? No, because they did learn. They were using the maternal emotional expressions to guide their behavior. They were just doing it in the opposite direction to what we predicted. But that wasn't to say that they weren't learning. They were learning. They were just using it in a different way from the other 2/3 of children. So they were learning. So we had to find some way of picking those two things apart, picking the direction of learning apart from the actual learning process. So that's why we split this into two indices. We have what we call the bias, which was, does the child follow the mother's instructions and go for the positive, or defy the mother's instructions and go for the negative, right? And that we think is more of a trait parameter. It had to do, we were analyzing this later on, what predicted that parameter. It's more of a trait parameter. It has something to do with temperament, really, actually. And so we separated that out. We call it the bias or trait parameter. And so how do we compute that? So if on in the panel C here, you see the child's responses trial by trial. So here, the positive responses are shown in blue, the negative responses are shown in red. The bias was taken as the weighted binomial probability score of all of these responses. And why wasn't it just a simple average? Well, we wanted to reward the children who sat through more trials because we thought that was a more reliable indicator of their actual abilities and preferences. So it was binomial computed weighted, but it was basically very similar to what we would see as a grand average across all the trials. But like I said, that isn't a good enough indicator of learning, because we can have a child who consistently goes for the negative response. And so, it would look like they were not, they're scoring very, very low on this bias score, but actually they were learning. So we wanted to pick these two things apart. So we had to then compute what we call the likelihood score, which was looking at the responses trial by trial. And to do this, we had to define the target direction, because this is how we pick those two things apart. So if we can see that the child has a positive bias, then their target is the positive object. So we would score 1 for positive responses and 0 for negative object responses. But if we can see that the child has a very low bias score, which means that they kept going for the negative, then we would flip the scoring. When the child touches the negative object, we would score that as 1. And when the child touches the positive object, we score that as 0. So the likelihood was corrected for or normalized by the child's bias so that we could take these two things apart. So when you see the 1s and the 0s for the likelihood score, that is whether the child is learning and going towards their internally selected bias direction or not. So it's already corrected for this a difference in bias across children. Okay, the last index that we wanted to compute, which didn't have this problem of bias, because this is a bit complicated to get your head around, was just pure reaction time. We didn't care what was the child going towards, just how quickly did they orientate towards it. So there were 3 indices of learning in this experiment, and we were interested in which connections, if any, predicted each of these different learning parameters. So here are the results. And you can see that basically there was a double dissociation. So let me just talk you through what these different plots relate to. The left column there are the connections just within the child's brain, just looking at the electrodes within the child's cap and calculating the PLV connections within the child's brain itself. The middle panel is connections within the adult's brain only. And the third panel there is connections across infant to the adult. So if you look at the axes, that's what they represent. Okay, so for the interpersonal one, the Y is infant, the X is adult. For this one, it's adult to adult, and for this one, it's infant to infant. So that's what the three columns represent, the locations of the measurement per se. The panels, the rows, are the learning index. So the top one was measuring likelihood, which is this, did the child learn or not, irrespective of their bias, learning or non-learning. And the bottom one was direction, bias, positive or negative. And what we did was we correlated these learning indices with the different strengths of connectivity in each of these different locations. And the voxels that are highlighted are the connections that survived very, very stringent thresholding and correction. So we corrected for a lot of things here. We corrected for movement artifacts, we corrected for infant inattentiveness, and we created a surrogate distribution just to make sure that none of these things were random connections. And we also took out anything that we could see was clearly driven by power correlations alone, because that is more likely to be driven by motion than otherwise, although it's not necessarily always the case, but to be safe, we took those out as well. So we did a lot of cleaning, or not cleaning, but thresholding to remove what could possibly be a confound in these correlation plots. And what you see here in red is what has survived that process. So as I've said, it's very clear that when you're looking at learning by itself, learning likelihood versus bias, this emotional trait, they are predicted by two very different sources of brain activity. The child's internal bias to select the positive or select the negative, that's predicted by a clutch of electrodes within the child's brain. So you can see the topography here is this kind of central group of electrodes, which is predicting whether the child on average goes towards the positive or the negative side. However, if you're looking at trial to trial learning, irrespective of bias, just did the child use the information from the parent to shape their response, then all the action is going on in the interpersonal domain. Nothing in the child's brain, nothing in the mother's brain is predicting that. It's the strength of the interpersonal connection from trial to trial, which predicts the child's response. So just to summarize that, that's the message which I've been telling you. The intra-infant connectivity predicts bias, which is a trait level parameter, whereas interpersonal connectivity predicts likelihood, which is a state trial to trial level parameter. And I didn't show on this plot, but actually when we looked at reaction time, we got a similar pattern of predictions in the interpersonal domains only in a very similar band of electrodes. So they would appear just around the edge of this plot that you see for the likelihood plot. But reaction time from trial to trial, being a state level parameter, was also captured uniquely by the interpersonal connections between the mother and the child. Okay, so that's just more of that. I think time is running out, so I won't go into too much detail there, but that's just showing you that learning over time is predicted by the interpersonal connection strength. So just to close the loop then, what is causing these fluctuations in interpersonal connectivity? Because it's clear that when the connectivity is strong, the child is using the information more effectively. So we looked at these changes in connectivity as a function of maternal behavior, and we looked at the usual suspects: maternal use of eye contact, maternal use of pitch. And again, these two things, if the mother increased eye contact in that trial, or she increased the pitch in that trial, then the interpersonal connectivity of the child also increased, and that was having an effect on the learning likelihood. So we've tried to now start to put this together in a model, because clearly, the interpersonal space is having a unique role, interpersonal connections are having a unique role in the learning process, but it doesn't mean that what's going on within the child's brain is not important. I think these are two halves and very complementary parts of the learning picture. So we need to take both into account. So what's going on here? So this is my very simplistic view at this point. We're getting more data, we're modeling this better. But I think we do have to think about these two steps maybe in the learning process, that yes, the child has a built-in bias, some priors, perhaps coming from their previous history, their previous experiences, their previous interactions with the mother, that are set points within the child's mind and cognition, about how they will process information when it comes in. But this is a trait level thing. It is how will I filter or process information when I receive it. But that by itself doesn't determine learning, because learning in a Bayesian model also depends on the incoming information at that time. So we have to pair that with the strength of the evidence that the child is being presented with from trial to trial. And there, I think, is where the interpersonal connection is a very good index of how convincing or how learning effective is the information that the mother is giving. So if the mother is doing a really good job of convincing the child with great eye contact, high pitch, strong emotion, and the child can clearly see that there's strong sensory evidence for learning, that drives the likelihood score very high because there's strong evidence. But that isn't the whole picture. That has to be combined back with the filter or the bias of the child to determine what the actual outcome is going to be. So I think that is probably the fuller picture that we need to look at. And in terms of mapping that back to classic psychology and how this interplays with the traits of the child, well, I would say that classic things like temperament and maternal sensitivity would affect things like the resting bias or the prior experiences of the child, the inbuilt filter, which you can see coming out in the child's interpersonal connections, strengths, and patterns. But the live evidence, what's happening in the moment from trial to trial, that is driven, as we've seen, very strongly by the ostensive signaling. What we're saying now, what we're seeing now, the sensory evidence that's being provided, that is changing the interpersonal connection from moment to moment, which is also an important part of that learning process. Okay. So I think that's all I want to say for now, but I'm very happy to engage later, but this is the kind of direction that we're taking things in to explain how things kind of fit together in the bigger picture. Right, I just want to conclude, because I know I promised more time for questions. I'm going to be quite quick about this, about how we might use this in the real world. So we are already developing coding schemes so that other people can start to use the information from the EEG and from the social interactions in a similar way. Because one of the big problems we have is this is interdisciplinary research. People who study play generally code interactions in a very different way that are not necessarily compatible with EEG analysis. So one of the first few things we wanted to do was develop coding schemes that would allow us to use, video footage that has been coded for, very richly for different kind of states of social interaction to inform EEG analysis. So without going into too much detail, this is one of those schemes that we've proposed for high dimensional coding of playful social interactions in a way that's compatible with dyadic EEG analysis. And then going forward, also thinking about what can this tell us about the state of the brain, the state of maturity of the brain. And also going beyond EEG, we thought dyadic EEG is great. It's showing these wonderful effects in communication and learning. But there are more domains that synchronize. Like I told you, there's eye contact, there's speech, there's arousal, physiological synchrony of arousal. There's gestural synchrony, there's speech synchrony. So why don't we make use of this rich picture of interpersonal interaction dynamics to help us inform the state of social health and also to predict the state of brain development. So this is now a bigger class of metrics that we're developing, which we're calling sociometrics, which is looking at dyadic interaction quality across multiple domains of interaction, but importantly, calculating indices of interaction. So not looking just at what the child's doing or just at what the parents doing, but looking at the interpersonal connectivity space across these different possible neurophysiological measures. And of course, in my lab, we've been very lucky to be funded to develop wearables and portable technology so that some of these things eventually can also be taken outside of the lab to inform home use or taken to the clinic for diagnostic purposes. And last but not least, since I do have research labs in 2 locations, and it is very much the case that a lot of what we know has come from a fairly small subset of cultures and research traditions, I think it's increasingly important to be able to do replication research across different locations. I couldn't end without flagging this up. But there are actually important cross-cultural differences in parent-infant interaction, particularly through maternal mental health, but also through just parenting practice, attachment styles, etc. And it's important to capture this in our developmental research. So this is also one direction that my labs uniquely are in a place to capture, where we can do exactly the same paradigms with the same equipment across 2 very different cultural cohorts. Okay, I just wanted to thank the people who have contributed towards the research that I showed you. So Sam, who works with me in UEL, our postdocs Kylie, Valdas, Lorena, and Stani, who is just graduating from a PhD as well. To thank the funders of the previous research, and to thank you very much for your attention. And I look forward to engaging further in any questions that you have. So thank you for your time again.